All right, you guys, since the internet was giving us issues yesterday and it kicked us off a couple times and we kept losing Zoom, I'm just going to re-record this whole lesson for you. So this is our last lesson for our chemistry course, nucleic acids and energy in living systems. So what are our nucleic acids? Nucleic acids are made of a phosphate sugar chain with nitrogenous bases connected to the sugar units. Okay, so here's an example. Phosphate sugar, phosphate sugar, phosphate sugar. Our nitrogenous bases are also what we call our nucleic acids. These are the same things, our adenine, thymine, guanine, and cytosine. And this right here is a model of deoxyribose. So this is what we're dealing with when we're dealing with deoxyribose here. And deoxyribose combined with a nucleic acid is how we get DNA, deoxyribonucleic acid. DNA uses 4 bases and forms a double helix by a specific ATGC pairing. Replication can only take place when the helix splits apart. So as you know, DNA is composed like a ladder, but the ladder is twisted. If you have a look over here, it gives you models of each of the molecules involved when we're talking about our nucleic acid bases. So here's our thymine and our adenine. These always pair together. Our guanine and our cytosine. These always pair together. And then when we're dealing with RNA, we have uracil. Now, uracil and thymine are those that pair with adenine. And in this case, when you're using DNA, you use thymine with adenine. And when you're using RNA, it's uracil with adenine. And if you look at their chemical structure, they're identical, thymine and uracil, except for this little guy over here. But this is not the part that is bonding with adenine, so it doesn't affect that bond. So here's how our pairing is done. We have 2 types of bases. Our thymine and cytosine are termed the pyrimidines, and our adenine and guanine are termed the purines. And these are where the bonds take place, this oxygen and hydrogen, this hydrogen and nitrogen, and then down here the hydrogen and oxygen, nitrogen and hydrogen, and oxygen and hydrogen. And they always pair this way in DNA. And then again, if you're dealing with RNA, this thymine goes away and it is replaced with a uracil. So here is a still graphic of how DNA replication works or transcription. So basically, you have this ladder, as we talked about before, and it's a twisted ladder right here, but it gets unzipped exactly like a zipper. This little helicase is what does the unzipping. And then you have these DNA polymerases that ride along the chain and they read this part of the chain, and they recruit the proteins to come in and pair up, not proteins, I'm sorry, the nucleic acids to come in and pair up with the appropriate base on the existing side of the chain. And this is how DNA would replicate. This is also how you would transcribe for RNA. It's the exact same process, except you'll be dealing with uracil instead of thymine. So the arrangement of base triplets on DNA encodes genetic information by dictating the synthesis of proteins. So what you're dealing with here is, here's a gene. Okay, this gene is this whole portion of this strip of DNA. And of course, your DNA is actually on chromosomes, right? And we haven't even gotten to that, but we'll talk about that a lot more next year in biology. So you have a gene, and the gene is composed of these little triplets. And if you remember from the lab we did last week where we did the DNA coding with the triplet bases, those are called codons. So when you put 3 triplets together, it's called a codon. And one codon is like a unit of the gene. So it's sort of like each of these codons is a word, like it was in the lab. And each of these words is part of a sentence, and that gene is a sentence. And it's going to code for specific protein production. It's going to do so by coding first for amino acids in a specific order, and then that will compose the protein. And the sentence by itself makes sense because it tells you a protein, whereas words on their own don't give you much. But without the context of other genes, that protein is just going to be a protein. It won't necessarily do anything specific. It's only within the context of the entire system that everything starts to make total sense. It's sort of like reading a paragraph. And over here is another example of base pairing, and you can see it's like a puzzle piece. In this case, we're dealing with mRNA, so we're transcribing, just like we were here, which means that our T's are going to compare with A, going to combine with A's, sorry, but our A's are going to combine with U's when it's on the mRNA side, because we don't have thymine in mRNA, we have uracil in. So this video actually shows how you get from DNA to protein.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So what are some technologies we have for DNA? All right, well, we have first this DNA fingerprinting or profiling. So this is what happens when you're dealing with crime scenes, and you probably have seen this on the news or whatever, or read about it, that somebody was caught because their DNA was left at the crime scene. Well, this is actually what it looks like. It's a block of gel, and you put a DNA sample in it and use a process called electrophoresis, which causes information in that DNA to travel along and then leave bands throughout the cell. So in this case, we have a victim, and this is our known, basically. We know this victim's DNA because we've taken a sample from the victim. And then we have crime scene DNA over here. So these are samples, perhaps it was blood or saliva or something else that was left at a crime scene. All right, so we have this sample here, and then we have this sample here. And in this sample here, you can see that it is identical to the victim. This means that the victim left his or her DNA at the crime scene, which makes sense. I mean, very often a victim's blood is found at the crime scene. But then if we're trying to figure out who this other sample belongs to, we have a suspect 1 and a suspect 2. And if you look, it's identical to suspect 2. Suspect 2 was also at the crime scene. And this is exactly how it looks like when it's done in a laboratory. Now, cloning. Cloning has become, well, hasn't, not recently, but it has become a hot button issue. It's been one for some time. So the first mammal successfully cloned was a sheep named Dolly. And that was done in 1996. And I was actually in college at the time studying this exact topic. So it was a very hot topic in my class, and it was really, really exciting to be learning about this in this process when it actually happened in live time. Pardon me. So what was done here? So we have this Scottish black-faced sheep, and she donated an egg. They say she donated. I don't know that she like voluntarily gave it up. I doubt that's how it happened. But anyway, an egg was removed from this Scottish black-faced sheep. And it went through a process called enucleation, where they remove the nucleus from the egg. And that's done physically, like through a surgical process under a microscope. And then we had a Finn Dorset sheep, and they're calling this a nuclear donor because the information in the nucleus of a cell from this particular organism was donated to the egg of another. So they didn't use a reproductive cell. They didn't use an egg from this sheep, or this was a female sheep. They actually went and used mammary gland cells. So they took cells from the udder of this sheep, and then they took that little cell and they combined it using electricity, actually, to the enucleated cell from this donor sheep. And then basically now you have an egg with a nucleus again, like you started out with, but the nucleus came from a different sheep. And the nucleus is where the chromosomes and the DNA are living. So now you've got a new organism. They waited till this developed in vitro in a lab to the point of blastocyst. It's a point in embryologic development. This is a cross section. So really it's like a bubble with a membrane of cells and then a little like ball of cells off to the side. And that was implanted into the uterus of a surrogate ewe, another sheep, another Scottish blackface sheep, not the same one that donated the egg and not the same one that donated the genetic material. And this blastocyst was allowed to mature through its process of gestation into a new organism. And it was born 5 months later. It's a lamb, and they named the lamb Dolly. Well, Dolly is a new and independent organism, but she is the clone of this sheep that donated the mammary cell. So this is basically like a twin to this other sheep because they're genetically identical. This was fascinating when it came up. It brings up a lot on the topic of ethics. What do you clone? What do you not clone? Do you clone entire organisms? There has been some advancement in technology in the last several years where they've been cloning particular types of cells to create organs or repair organs. And so it's a very fascinating field. Recombinant DNA has lots of applications, including making more pest-resistant plants, developing vaccines, creating insulin and growth hormones. So this is very often a technology used in the pharmaceutical industry. This right here, my friends, is a mitochondria. Mitochondria we call the powerhouse of the cell, and that's if we're referring to animal cells, because the powerhouse of plant cells or other, well, we'll say plant cells for now, we'll speak generally, is the chloroplast. So that's because the process that creates energy or stores energy in plant cells or photosynthetic organisms in general is photosynthesis. And in that process, you're going to take in carbon dioxide and water and energy from the sun, which is what you see trees doing every day. Well, you don't see it, but you know they're doing it. And that is converted into sugar and oxygen. The oxygen comes out as waste, and the sugar is stored. So green plants use solar energy, carbon dioxide, and water to synthesize glucose during photosynthesis. Cellular respiration is the exact reverse opposite process. So you're going to take stored sugar and oxygen, and it's going to be converted into carbon dioxide, water, and energy. In this case, the carbon dioxide and water are given off as waste, and the energy is expended. So if you eat a salad, you're getting sugar that was created during the process of photosynthesis, and that sugar is here, and you're going to combine it with oxygen that you inhale. You're going to exhale carbon dioxide and water, and you will have energy. Also, say you're not eating a plant, say you're eating a steak. Where is the energy coming from? Well, that steak at one point was a cow that ate grass. And if you follow the food chain back far enough, you're going to end up with a photosynthetic organism almost every time. So the reverse of photosynthesis is respiration at the cellular level, which is glucose broken down into carbon dioxide and water. Energy is harvested by the process of production of 38 ATP ions per glucose molecule. ATP ions, we're going to look at in a minute, but it's adenosine triphosphate, and that is basically our little unit of energy. So here is a little diagram showing you what happens inside a chloroplast. That's the photosynthesis process. And mitochondria is where the cellular respiration process occurs. And both of these are organelles that exist inside of cells. So here's a graphic of cellular respiration, way disproportional. Obviously, this is out of proportion because it's more like an infographic, but it's discussing that ATP fuels life's processes, motion, synthesis, and transport. So here is a giant mitochondria inside of this little cell, which, like I said, it's not proportional, but when you get glucose, it breaks down into pyruvic acid through a process called glycolysis, and that's going to yield an ATP. And then you've got two other options going on here. One of them is a Krebs cycle that's going to produce ATP. Another is electron transport, and that's going to create more ATP. And these are your little, this is the chemical basically that's going to fuel all of your life's processes. And here you've got your carbon dioxide and water being given off as waste. So that was our energy and living systems. Now I have a little video for you that goes along with your homework regarding HIV medications and HIV as a disease. So we'll watch the video first.
It has been 20 years since Benta Gola first found out she was HIV positive. At the time, she was only 27 years and 6 months heavy with her fourth-born child. But it has been a long walk to living freely but positively due to the stigma associated with the disease.
We were pointed at, we were talked about, people still talked about us, and it was sad. For some time, I told my counselor I don't have to go for medication.
A situation that a majority of the 1.5 million persons in Kenya living with HIV have experienced at one point in their lives. But a situation that could change given the latest approval by the US Food and Drug Administration of a two-shot combo called Cabenuva. This becomes the latest innovation in treating what has become a chronic, manageable, but still incurable disease. An innovation that gives hope of reaching groups that have found a hard time sticking to treatment, people with mental illness and those that have substance abuse problems.
It's going to make it easy for people to actually take their medication. And hopefully, I think it's going to improve the quality of life for people living with HIV.
That will be good because I'll organize myself. I know such and such a day. I'm not only going for injection, then I come back. Yeah, so people will not be hiding themselves that she's just been tested. She's not telling anyone. She's not going for medication. People will come out.
The injectable known as Cabenuva is a combination of two drug formulations, cabotegravir, which is a new drug, and also one described to be able to prevent one from being infected with HIV, and rilpivirine, a drug that has been used before in the treatment of HIV-positive persons. The drug packaged together will be given as separate shots once a month.
And we've come from far because we came from a time when people take up to 18 pills a day. And, there will be challenges because they have to take some of these pills with food. So there were challenges getting food.
At times, I'm so much exhausted. Before I watch 9 p.m. news, I'm already asleep. So at times I forget. So it has been a hurdle.
According to the FDA press statement, an approval has also been made for Vocabria, which is the cabotegravir tablet formulation, and which should be taken in combination with oral rilpivirine for 1 month prior to starting treatment with Cabenuva. This is to ensure the medications are well tolerated before switching to the extended release injectable formulation. For Dr. Peter Cherutich, a public health and HIV/AIDS specialist, the cost of the medication will be expensive in its initial stages, just like other innovations. However, given the commitment of donor funding, the drug will be made affordable over time.
First, for you to get to injectables, your viral load must be well controlled. Of course, there's still challenges on children and dosing, but that will come later on.
At the moment, a majority of the persons living with HIV are taking one pill daily, which is a combination of 3 other drugs. And while the injectable will replace the daily pills, a HIV-positive person will be required to take other medications that will prevent them from opportunistic diseases. Raquel Muigai, Citizen TV.
Your assignment was to research to find out more about HIV, identify the kinds of cells the virus attacks, and describe how the viral infection leads to HIV/AIDS. Find out more about retroviruses, how are drugs used to combat infections caused by these retroviruses, what is Cabenuva, and how does it work? Oh, does it work? Look at that. I don't think anybody caught that yesterday. So this basically covers what the video just saw, and HIV attacks white blood cells called helper T cells. And that HIV attacks white blood cells called helper T cells. Helper T cells help regulate activity of many other blood cells that destroy invading pathogens. Pathogens are basically anything that doesn't belong in your body. This response to pathogens is called immune response. It enables humans to fight off bacterial and viral diseases. Destruction of many T cells weakens immune defenses so much that the body fails to ward off common infections. This condition results in AIDS. And there is a part of your answer to number 2 there. And if you had something including tumor viruses or leukemia, also feline leukemia, people may have mentioned, even though it doesn't affect human beings. And I have one more video for you, and this one is on viral replication. And this has probably been an important topic to cover this year, considering we have been dealing with a viral pandemic. But this will explain with some great graphics exactly how a virus replicates.
Viruses. Viruses are infective agents too small to be seen with a microscope. They all have a similar structure, being surrounded by a protein shell called a capsid, which protects the viral genome. Inside this capsid is either viral DNA or viral RNA. Also present is the viral envelope, which is covered in attachment proteins. The viral envelope is made of lipoproteins. So how do viruses reproduce? Well, they don't quite lay eggs, that's for sure. Viruses don't have a way to reproduce alone. Instead, they need to hijack a host cell. To do this, they approach a cell. That attachment protein we mentioned earlier interacts with specific receptors on the cell membrane. The protein and the receptor are complementary. The contents of the virus, such as viral RNA and the reverse transcriptase enzyme, are then injected into the cell by endocytosis. The reverse transcriptase enzyme copies the viral RNA, forming double-stranded DNA, which is then transported to the nucleus. The viral DNA is then integrated into the host DNA, and the new host cell assembles new viral DNA and the proteins necessary for the capsid and envelope. The new viral DNA and enzymes then leave the cell by exocytosis, constructing a viral envelope from the cell membrane of the host cell. Eventually, this will kill the cell, while the virus, well, they're free to infect other cells, and the whole cycle begins again.
So one more thing I wanted to discuss with you was the issue, one more thing I wanted to discuss with you was the topic of DNA phenotyping, because this is pretty fascinating and it's a brand new emerging technology. So there's a company called Parabon Nanolabs located in Northern Virginia. And since 2018, they've helped police solve more than 120 cases with their genetic genealogy and then DNA phenotyping methods. And DNA phenotyping is a fascinating new technology. So what they do is, in this case, for example, they have an unidentified person, a Jane Doe, a body, perhaps, and perhaps this body is at a point where it's degraded to the point where you can no longer identify facial features, or maybe it's completely a skeleton, or maybe you only have a part of a body, like a limb or a tooth or a bone. So what this company's technology, hold on, I'm filming, so I'm waiting for you to, are you good? Yeah, I was filming, so I've paused because you were stomping down the stairs. Do you need?
Oh, sorry.
No, you good?
Yeah, I'm good.
Okay.
Because
All right.
I got calls for most of the day.
All right. So what they've done here is they can take DNA sample from that Jane Doe, which they did here in this case. And they have, I have a video, one more video, sorry, where the lady that works for this company explains how it works. But they have a massive database.
Digging ditches.
They have a really massive database containing information regarding how genes are expressed. So if you have a specific gene, it'll tell you how it's likely to be expressed. And they can use percentages, for example, and say, in this case, there is an 83.9% chance that this person's hair color fell right in this range. And they can also tell you what you're not going to find. Like in this chance, they're saying there is almost no chance that this victim had freckles. And then they combine all that data in their computer system and it spits out a composite sketch, but it's a digital sketch. So it's kind of like the sketches that you may have seen before on TV where they do like a crime scene sketch of a potential perpetrator, or sometimes they'll do them with victims. And they've also in the past done the thing with skeletal remains where they put those little pins on their skull and then they build clay around it and kind of model what the face should be. So with DNA phenotyping, they create this digital image. And then this digital image allowed investigators to put it out and say, hey, we found somebody who looks like this. Is anybody missing her? And lo and behold, this is the young lady who matched the sketch. It's pretty close. They did a really, really good job. And once they put a face to this body, they were able to solve their crime and find out that this man right here, he was the one accused of, it says, killing her and then dumping her body in the woods. So now they have the information they need to move on and solve the crime. So this was a Jane Doe case. Now in this situation, it's a little different. This situation, you have a perpetrator who left DNA behind at a crime scene. This is the victim over here. So they've done DNA phenotyping with the perpetrator's DNA to try and put a face to that DNA to say, who was this guy? Well, they don't have a way of telling how old the perpetrator is necessarily. So they put the perpetrators in general, when they construct these faces or victims, they put them around 25 years of age. I'm sure there's a good reason for that, but I'm not positive why. So in this case, they created this composite from the DNA around 25 years of age. And they can tell us here, you know, you've got very low chance of freckles, your hair color is going to fall in this range, your eye color is going to fall in this range. And this is the actual perpetrator. And so you can tell that these faces are still pretty similar, but it's definitely difficult when this guy is much older than the composite. However, if you knew him when he was 25, you'd probably go, oh, there's Bob again. Oh, I always thought Bob was sketchy. And it allows them also to narrow down their field. They can say who they are not looking for. In this case, they are not looking for someone with blue eyes. They are not looking for someone with lots of freckles. They are not looking for someone with darker skin. And that really does help them narrow down their pool of suspects. So in this last video, she is going to be explaining to you a little bit more about the technology of DNA phenotyping and how it works.
I'm Ellen McRae Graytack. I'm the Director of Bioinformatics at Parabon Nanolabs. And we do DNA phenotyping, so predicting a person's appearance, ancestry, all that stuff just from a DNA sample. We start from what we call genotype to phenotype data. That just means a big database where on thousands of samples, we have both information about their phenotypes, like say eye color. We have, are their eyes blue, green, brown, and we also have genotype SNP data. So we have got about 1 million SNPs on each person and their genotype. So that's their AA or are they GG at each site. And what we want to know is which of those SNPs is significant for eye color. So we basically look through at each individual SNP and score how well does this data associate with the phenotype data. If people who have an AA at this SNP always have blue eyes, well, that would be a really strong association. It's never that simple, but that's what we're looking for. But the thing is, it actually gets much more complicated than that. If you look through the literature in genomics, you find what they call missing heritability, which means there are these traits that we know are genetic. They pass through families. They're very clearly heritable. But when we look across the genome, we don't actually find all the information to explain all the variation in that phenotype. So that's missing heritability. We're only explaining some of the heritability. And one idea for what might be missing is actually interactions among SNPs. So you might have a set of 5 SNPs that each of them individually has no effect on phenotype. But if you look at all 5 of them together, there are certain combinations of genotypes that have a significant effect. But if you've got 1 million and you need to look at 5-way combinations, that's way more than you can ever test. I mean, you'd be doing calculations till the end of the universe. We don't have time for that. So what we've done is we've developed software that's called an evolutionary algorithm or a genetic algorithm. And so what it does is it starts from a random population, and we're still doing scoring. We're looking at this combination of SNPs and scoring how well does this predict that phenotype. If it scores well, it gets to survive and breed and mutate, and we get to look all around it at other possible combinations of SNPs. If it scores poorly, then it dies off. It doesn't get to get searched around. So in that way, we're trying to explore the important parts of the space while not wasting the rest of our lives looking at the entire space. So that's how we're trying to discover SNP associations that haven't been found before. So that's just the data mining. Then we build it into a predictive model. So we take those SNPs that we found and we say, okay, we know that that AA genotype is highly associated with blue eyes. So in our predictive model, it would say, if I see AA, I'm going to give a higher probability to blue eyes. That's basically what the predictive modeling is. So for kinship, we also started from scratch. It's a similar approach where we also have a database, except now instead of eye color being our phenotype we want to predict, it's I have these 2 genomes and they're related at this distance. They're 3rd degree relatives, for example. So we're taking the same approach, except instead of looking at each genotype, now you have 2 genomes you have to look at. And so what we're looking at is how similar are these genomes? And can we use that information to predict the relatedness? And so we've got a database that goes out to, there are actually 7th degree relatives in there, as well as many unrelated pairs. And we're using this to predict some 2 people's relatedness. And the value of that for forensics, it's twofold. It could be maybe the perpetrator of this crime is related to another perpetrator, a victim, a neighbor, you could test any pair of people that you suspect might be related. The other possible use is if you have remains and you don't have an immediate relative to test, you don't have a parent or a first sibling, but maybe you have a cousin and you suspect that it might be this person's relative. Well, now you can test their DNA and see, does it match the relationship of someone a 3rd degree relative? Well, the challenge with faces is basically to describe a face in a way that can be predicted. So from our point of view, if you can measure something, you can predict it as long as there's some underlying genetics. So what we do is we take the face and turn it into just a combination of variables. How wide is the face? How long? How angular? How big is the nose? All of those can be turned into numbers that can then be predicted. So we're looking exactly in the same way that we did with eye color. We're looking for what are the genes that are involved in this? How can they be used to predict what this person's face like? This specific person whose face is somewhere along all of these variables, maybe their face is wide and short or long and narrow, all of those things are somewhere in that face space, what we call it. We just need to be able to predict those variables. So when we use this for casework, what we try to do is emphasize both what we can predict and what we can exclude. So we don't just say this person has blue eyes, because a lot of the time, maybe this person has dark blue eyes. They're sort of on the line between blue and green, and it's very difficult to predict or even know, looking at them, are their eyes blue or green? And so that is going to hurt our confidence. So whenever we report a result, we say this person has blue eyes, and we have 75% confidence in that, for example, because maybe their eyes are green. However, maybe we can also say with 99% confidence, this person does not have hazel, brown, or black eyes. And that's what's really important for investigators. We can tell them you don't need to look at the people in your suspect list who have dark eyes, because they really do not match this profile that we've come up with. And there's only maybe a 1% chance that this person has dark eyes versus a 99% chance that they have blue or green eyes. So that's what we try to focus on, is how can you use this information to narrow a list of possible matches. And so we've been using this in casework. We've done several dozen cases at this point where investigators come to us, say this, I've had this case hanging over me for years. I have no witnesses, and I just don't know who to look for. If they still have DNA from that case, we can do our genotyping assay, analyze that, and tell them, okay, you're looking for someone who matches this profile, and this vast majority of the people that you see walking the street are not going to match their profile. So you need to focus in on this smaller subset of your possible matches. But what we really want to see in the future, as this is getting out there, like I say, now it's mostly that oldest, worst cold case. But imagine now a crime occurs, and before you spend 5 years looking at every person around, if you narrowed it down to 5% of the population, suddenly, okay, I'm only looking for this blue-eyed person, and that's really going to change the efficiency of these investigations, and that's what we're looking for going forward.
So that's it, our last lesson of the year. I'll see you guys next week for review, and then we'll follow up that with exams and projects.